We are really honored today by our guest retired just recently from the Mayo Clinic, Dr. Virginia Miller. She is a leader in the field of sex differences research like few others out there. And she's going to teach about basic concepts of sex as a biological variable. And we talk about it all the time, but to have somebody actually speak to us about it is of great benefit to all of us. Before I begin, I have a few announcements for the Ludeman Family for Women's Health Research. We're grateful to the many donors and supporters of our center who have made it possible to offer supplemental grants. And thanks to you who submitted supplemental grants, we're going to be evaluating them in pretty short order, and we'll get back to our recipients as soon as possible, which will be soon. Our Center of Virginia, our board contributed, and the center put out money so that scholars could apply for supplemental funding, given the hardships that COVID brought to everybody. We're going to be sending out a survey following this training. Please complete the survey as it helps us to plan for future topics. As we begin to transition back to campus, we're going to have an in-person event. All right, in mid-September at the Anschutz Health and Wellness Center. Please look out for an invitation. You will all be invited, so that'll be fun. You'll actually see people. It's always so shocking to actually see people, and they don't have masks on anymore. Mark your calendars, please, for our annual community event, which will be on October 5th. Dr. Jennifer Miras, who is a renowned cardiologist, African-American cardiologist from the Northwell Health Center, talking about taking care of your heart. And she is going to talk about women's heart health, but she's also going to talk about men's heart health and focus a little bit on diversity and how racial and ethnic influences affect the heart. So I think it'll be really great. We're planning to have an in-person event at the DCPA as well as a live stream event. And, being that things are not very clear to us entirely, we're going to, go with the flow a little bit, but we're working on these events. There will be for sure the live stream, and we think there will also be a live event. So that'll be exciting. Our next researcher training is July 9th at 8:30. Tim Lockey of CCTSI will speak on grants administration basics and how to write a budget, how to organize a grant. He gives an incredibly useful talk on this subject. Now to turn to Dr. Virginia Miller. During Dr. Miller's stellar career in women's health and sex differences research at the Mayo Clinic, her research has included both basic research and also there have been clinical research efforts. She is focused on how sex steroids and conditions unique to women, such as pregnancy and menopause, affect cardiovascular health and cognition. She was the principal investigator of the Mayo Clinic Birch grant. As I am here, she was there and brought a lot of energy and good thought to that process. And I'm sorry to lose her as a Birch colleague because she's one of my favorite Birch colleagues, for sure. And she's authored over 300 original publications and reviews. She's a very, very well-funded scientist throughout her career. Her awards have included the Bernadine Healy Award for Visionary Leadership in Women's Health, the Women's Day Magazine Red Dress Award, the Paul M. Van Houte, am I pronouncing it right or wrong, Virginia? Van Houte, thank you. Named lecturer in vascular pharmacology. He was a very famous vascular scientist. And the Walter B. Canada Award from the American Physiological Society, another very prestigious award. Her professional service was amazing and includes her coming today, which she just told us is going to be her last official talk, which is horrifying to me, but I'm happy for her as well because she's a great speaker. So without further ado, I'm going to introduce my esteemed colleague, Dr. Virginia Miller.
Okay, thank you, Judy, for those very kind words. And indeed, it is a pleasure to be here today. And I wish I was in person. I've been to Colorado many times to your center, and it's an amazing place. And Judy has done such a wonderful job of organizing the support that it now is a named center with a legacy that's going to carry it forward well into the future. So she asked me to start off this series on sex and gender with some basic topics. And although I've been giving these similar talks for many, many years, when I prepared, I figured, is this anything new to anybody? Because I've done it so many times. but I'm going to go through it again. Some of it you may know, some of it you may not. And I think because it's kind of a workshop kind of thing, although I can't monitor the questions or comments, someone's going to be doing that for me. So we feel free to kind of interrupt as we go along. And hopefully I can get some interaction from you, even though it's so difficult with the Zoom. I miss seeing people and having hands raised in the audience and so on and so on. So I don't have any conflicts, financial conflicts of interest anymore, being retired, but I did list my previous funding sources because most of the work and concepts that I'm presenting today were developed while I was funded by these agencies. So what we're going to do today is pretty simple. Let's keep it simple. We want to have you be able to articulate the rationale for sex-based research, list factors and mechanisms contributing to sex differences, differentiate sex from gender, and identify key components of a research strategy so that you can continue sex and gender research into the future. So let's get started. Why should we consider sex as a biological variable in research and clinical practice? Well, this is fundamental because research is the driver for education and a cutting-edge clinical practice. You can't have new clinical practice without the research to support it. And when we think of a sex-based approach, it really is the essence of holistic approach to patient-centered care. Because if I look at all the differences or the people that have joined this conference today, the biggest difference I see is whether or not you're male or female. And so it's really basic to how your physiology and disease risk is. And you'll see that in a minute. NIH took a while to get onto this concept, and it seems like it was just recently, but it was really 7 years ago when they came forward with a proposal to balance sex and research for transparency and reproducibility, which is a little bit disappointing because it didn't come a concept of fundamental physiology. You know, reproduce your results is one thing, but applying it to be able to apply it to the health of the nation, which is really what NIH directive is to improve the health of the nation, not to build a better mouse, you know, is fundamental to that. But for transparency and reproducibility, it's good enough to get us started. So let's just review why it's so fundamental. Let's take a look at the human karyotype. And as you can see, the basic difference between males and females are these sex chromosomes, 2X for the female and an X and a Y for a male. And when you think about this, in the nucleus of every nucleated cell, there are these sex chromosomes. So of course you can conclude that every cell has a sex. So with that basic assumption, why would we forget about it? And we have to consider it when we do cell culture experiments, the effects on tissue, and of course, organ and integrated physiology. So at the basic level, at the genetic level, and we do these, interested in the genotypes and gene variation, we should also be interested in the sex chromosomes. All sex differences result from the imbalance of the X and the Y chromosome. And this drawing is compliments of Art Arnold, who has been studying sex differences from the genetic perspective for a long, long time. And these effects are non-gonadal. That is, that you see these sex differences effects when there are no gonads present. And that's the most obvious then would be isolated cells. These are the sex chromosome effects. So what are they exactly? Well, let's look at the number of genes that are on the sex chromosomes. And you can see that I just summarized these from a variety of summaries. And we see that all the major systems are represented in some degree, especially for cancers, cardiovascular disease, cell regulation and metabolism, endocrine, hematology, and one is all in our minds right now is immunology is related to the coronavirus. And when we look at the Y chromosome, there's few genes on this chromosome. Of course, there's some that direct the development of the sex organs, but some have been associated with hypertension, and another group has been associated with coronary artery disease, especially in terms of immune signaling and inflammatory processes. And when we think about this, and we just think of what you've heard in the news lately, about the coronavirus vaccine causing more adverse effects in young men in terms of myocarditis and endocarditis than in young females. So these ideas that these genes on the Y aren't important, they are, and some of them actually do regulate gene function on the autosomes. So if females have 2 X chromosomes, there would be a dose effect of the X chromosome compared to males. To compensate for that, one of the X chromosomes is inactivated by methylation. So in females, you get an X chromosome from your mom and one from your dad. Doesn't matter which color they are here. But one being inactivated then makes the expression of the X chromosome comparable in females and males. Now, the question can be, yes, there's just one there, but are they comparable? So think about it for a minute, and what would make this different between the males and females in terms of genes that are expressed on the X chromosome that would in males compared to females? And I can't see you, and I can't know if anybody's raising your hand, so I'm just going to carry on. So when we think about genes that are on the X chromosome and one is inactivated and some of them are not, we can think about the androgen receptor. The androgen receptor is on the X chromosome. So any genetic variation in this androgen receptor is going to be more prominent in the males than the females. The androgen receptor is one that is not inactivated. So if you have genetic variation here and differences in expression of the X chromosome, those are all going to show up in the male and not so much in the female. So that would be androgen insensitivity, which would be resulting from that genetic variation. So with X inactivation, you have a mosaic gene expression in all the cells, because this X inactivation is random. And this is a calico cat. Calico cats are always female. And this is a good example of how that mosaicism, in this case, coat color, is expressed due to the inactivation of one of the X chromosomes. Now, coat color is kind of interesting and it's kind of permanent, right? But what does that mean in terms of our physiology or design of experiments and the subsequent consequences of this inactivation when we're studying systems and sex differences? I'm going to go briefly over this experiment, which was published some years ago, but I think it's quite clever, and it emphasizes the importance and what we don't know about this mosaicism. So in this particular experiment, what they did was to tag the X chromosomes with fluorescent material in the paternal and the maternal animals. And so the resulting siblings show this different fluorescence depending upon whether or not the X from the mom or the dad is being expressed. As you can see, it doesn't matter whether one's green or red, but you can see that there's a very interesting and non-homogeneous pattern of this expression in these siblings from one litter. Here, one is almost all expressing from one parent compared to the other. And then there's this whole variation in between. So what does this mean when we look at specific organs? Remember, every cell has a sex. So if we look at heart muscle, for example, you do see this kind of mosaicism pattern. And when you take a slice through this muscle, you can see that some of these cells expressing one of the X chromosomes compared to the other are actually kind of clustered. Now, we don't know exactly what causes this or regulates this or what this means overall in gene expression. But when you think about this in a female organ that you're studying and whether there's paternal genetic variation or inheritance from the dad, this is going to create a variability in your female population or the cells that you're studying, especially if you're doing isolated cell culture, if you're deriving primary cell a culture of primary cells, and typically it's not evaluated whether or not those cells are expressing maternal or paternal X. This is the heart. This is one of my favorite tissues to study as a cardiovascular physiologist. But the other thing we're also interested in is the brain. This is the coronal section through a rat brain. This is the cortex up here, and these are the pyramidal cells of the hippocampus. And again, you can see that the sides of the brain are showing very different expression of the paternal and the maternal X chromosome. And when you look at the pyramidal cells too, these does not show a, sort of a random expression of one X compared to the other. So you kind of wonder how this translates out into human behavior or of characteristics you might inherit from your mom or your dad that may be related to the X chromosome, and also cognitive functions and ability to, if you inherited some particular characteristic of your desire for architecture or mathematical ability or whatever, you kind of wonder how that's associated with the expression of these structural characteristics that may happen from your mom or dad in the brain. So when we think about every cell having a sex, then we think about that as influencing the regulation of ion channels, biomarkers, importance on stem cell transplant, whether or not they're autologous or heterologous, or transplant female organs into men, males, male organs into females, and so on, how those factors are affected because of the presence of the X and the Y chromosome. Oops. Okay. Went ahead. But the sex chromosomes also dictate the development of the gonads, and the gonads produce sex steroid hormones, which cause organizational effects, which are non-reversible if those hormones go away, and those specifically relate to the structure of the reproductive organs, or the activational effects, which can be reversed if the hormones are present or not. And good examples of those are the presence of the hormones during puberty, and the loss of hormones during menopause in women, and the hormonal changes which are associated with pregnancy. So let's talk about these hormones for a minute in terms of what we need to think about when we design our research studies and using sex as a basis of research design. So keep in mind that the sex steroid hormones are produced from cholesterol, and the estrogen is produced from aromatase. So this is an enzymatic reaction. These increase with puberty and decline with aging. And both men and women have both hormones, testosterone and estrogen, but in different concentrations. Now take a look at these pictures for a minute. And what might strike you or you find interesting? I can't get any feedback, so I'm just going to have you think about it in a minute. And I'm going to sort of prime the pump here by saying there's three important characteristics of these depicted here that you need to consider in your experimental design. First of all is the sex, whether you have XX, XX, or XY chromosomes. The second thing you need to think about is the age of the individual. The third thing you need to think about is the hormonal status. So you can see that, I have pictures here of individuals from different races and ethnicities. Some of these things we can't control. We can't control our sex at birth. We can't control our race, and we can't control how we age. So these characteristics and the fact that you have the hormones changing throughout age really need to be considered when you're designing your study for the question that you're asking. Do you need to study younger individuals, or the results from the younger individuals comparable to what might be of individuals that are reproductive age? And what about aging? Do factors that affect risk for disease or treatment, are they the same in individuals of reproductive competence compared to individuals who might be aging, and women, especially when they pass menopause? So those kind of variables you need to think about in terms of how you're designing it. Well, you're lumping people together with an age range in terms of your inclusion criteria, your exclusion criteria, and really have a solid scientific reason for why you're including that age group or not. And if you include a wide age group, what are you doing to your results in terms about application to the general population? Would it be applicable to the general population if you don't have sufficient numbers of individuals that are male or female, or of each age group, to be able to identify whether or not your treatment effect or your regulatory mechanism is actually effective? So how do these hormones affect life? Well, I have this wonderful picture here from the Journal of Applied Physiology, which was in a series in 2003. And when we look at disease risk, and you can just sort of picture this here for these individuals, you know, you have disease risk associated with puberty, of course, for women, hormonal contraceptives, and these are sex specific, pregnancy, surgical induced menopause, and natural menopause and hormone treatments. These are things that are not typically considered when you on intake forms or risk factors for clinical studies. And I'm encouraging you to think about that and begin including them as potential confounders or variables in your outcomes. So what does this mean in terms of how the steroids, sex steroids, and the genome affect physiology and pathophysiology? Well, when we think about genomic regulation within a cell, these are factors which are dictated by the genome, and the X and the Y sex chromosomes are included in that. These affect, gene transcription, translation, and affect all areas of cell function. If you put on top of that the effect of hormones, we have non-genomic regulation, which some of these hormones can affect the function of ion channels, surface membrane receptors for cardiovascular folks, nitric oxide, intracellular signaling pathways. And these do not require the presence of the genome. However, you can have an indirect effect of these non-genomic actions because some of the activation of these cellular signaling pathways can affect gene transcription. In addition, the hormones themselves combine with intracellular receptors, migrate to the nucleus, and combine with response elements on the genes and have a direct genomic effect. So when we talk about the sex differences in cells, we've got the genomic effect of the X and Y chromosomes. You have the genomic effect of the sex steroids influencing the gene regulation. These usually take quite a long time. Genomic regulation, is over, hours in a day. With a non-genomic regulation, with especially regulation of ion channels and nitric oxide, and some of these intracellular signaling pathways can occur within minutes or less. So when you have a fellow from our Birch program or one of our training grant workshops who said, I'm not interested in sex differences, but I'm interested in fundamental mechanisms of cellular regulation. For example, I want to know what regulates large conductance calcium activated potassium. So what's wrong with this statement? If you're interested in regulation, a mechanism of regulation, and before we just talked about that the sex chromosomes and the hormones affect regulation, you can't make a statement like this because fundamental mechanisms may be that the mechanisms of regulation are different in males or females. they have hormones that may compensate for these differences to make it look like the channels are working similarly, but by different mechanisms. So you have to consider the regulatory pathways. And the question is, are they the same? And if so, how are they are different? And how are they regulated by hormones or not? So you can't get away from the idea of fundamental mechanisms without considering somewhere in the back of your head that they may be different between males and females. They may be regulated by sex hormones. And you need to address that when you're designing your study and in how you interpret your data. So in essence, to summarize this beginning part of my talk here, when we think about the biology of sex differences in health and disease, you have the chromosomes, which direct all bodily functions, okay? And they're going to influence the development of hormones, brain networks, neurotransmitters, organ structures, response to stress, fat distribution, immune responses, homeostasis, blood water, metabolism, all of these things, which I know are of interest to investigators at the center there in Colorado. I've heard the talks, especially with metabolism, fat distribution, and regulation of intracellular pathways. So let's look at a couple of examples. And I'm going to use this one from kind of a pharmacological point of view. How might sex hormones interact with medications and metabolic pathways? Well, there's four major ways. One is that they can compete with transporters, and statins definitely do that. They can interact with other receptors or compete for the same type of receptor. They can compete with the enzymes, and they can alter gene transcription. So when you're looking at the effects of medications, and is it important to think about the hormonal status of the individual and whether or not these medications are going to work the same in men or women, or you need the same dose, do you need the same formulation, or the pharmacokinetics and pharmacodynamics the same? And there are many instances where that is not the case. And I must say that, I know that some of the clinical studies for coronavirus, when I went on the website to look for that, did enroll individuals of both males and females of different age groups, right? That's how the studies were designed. Yet when I went over to the Civic Center here in town in the Minnesota Lottery to get my shot, we're standing in line and they're all lined up and bang, 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 we're getting the exact same dose, whether or not you're somebody my size or somebody that is a 300-pound male. Now, I'll just leave it there. Does that make sense to you? And I know if in a pandemic, you need to get it out there and infuse everybody, but in the long run, is this how we want to treat everybody that comes through the door and for chronic conditions? And I think you all know the answer to that. Another example in terms of cardiovascular science is that we have an integrated system here where we have some autonomic nervous system affecting the heart, affecting the blood vessels, affecting hormones that might be coming out of the adrenal gland and kidney function. And if we take a look here at what might be happening at the nerve ending, we can see that the characteristics on the Y chromosome regulate tyrosine hydroxylase, which is the basic formulation for tyrosine and dopamine, which is a precursor to norepinephrine. So you have clearly regulation of this hormone system by estrogen and testosterone, the presence of the Y chromosome. Furthermore, you have different distribution of the adrenergic receptors on the end organ, where you have some of these which are regulated by estrogen, and the beta receptors are one of them, and the alpha-2 receptors are another. So when you think about cardiovascular regulation, is it going to be the same with activation of the autonomic nervous system between males and females? And does this have any relationship to development and sex differences in the development of hypertension? And we know men develop hypertension at an earlier age than women. It also affects how you look at factors when you're doing gene mapping. And this was a study early on I made with some of my fellows at a seminar course who were interested. They were cardiovascular people. They were interested in heart failure with preserved ejection fraction, or HFpEF. And when we put sex as a variable for all these other systems that we knew that were regulated, clearly the sex is different. It affects estrogen and testosterone. And you can see that there's connections to molecular pathways that affect cellular function and organ function. And so considering sex in any of these mapping experiments is critical in order to really move science and medicine forward. I'm just going to talk a minute about just some things about the transgender physiology for a minute. So in females with XX chromosomes, their primary hormone is estrogen, but for transgender procedures, these are given testosterone, which give activational effects. Remember, those which are present when the hormone is there and absent when they're not, for a more male phenotype. And here is a good example of differences between organizational and activational effects of the hormones, because even in these individuals who go through this hormone therapy, their sex organs are present unless don't change unless they are physically removed. The good difference between activational and organizational effects. And I think when we think about the transgender population, there again, as we talked about the hormones, there's three characteristics here you need to think about. What is the age in which these hormones were given or changed? What are the formulations? And what is the dose? All of these are going to affect the physiological and cellular mechanisms in terms of disease risk in response to treatment. And so there's questions about whether or not individuals should be included, trans individuals should be included in clinical trials. And I would propose that it does not do the study any good or the actual transgender population any good to be included in groups that do not have this very unique combination of chromosomes and hormonal variability. So why does sex matter in physiology and pathophysiology? It matters because we have sex-specific chromosomes and hormones. And as a result, there are sex differences in incidence, prevalence, etiology, symptomatology, response to treatment, morbidity, and mortality for almost any disease that you think about. There's some difference in one of these. And so I would, think about your own research and think about what you know about one of these factors in terms of a sex difference, and how does it matter in terms of regulatory physiology, response to the environment, or health and disease. So far, we've talked strictly about sex, biological sex. So what is the difference between sex and gender? Well, the Institute of Medicine definition of sex as being male or female according to reproductive organs, and importantly, functions assigned by the chromosomal complement, that is the XX and the XY chromosome, the sex chromosome. So sex is biological. Gender refers to cultural attitudes and behaviors. So one easy way to say it, sex is biology, gender is everything else. So what does that mean in terms of how we have this big emphasis on gender these days? Actually, when you look at the history of gender, it really comes from initially from the designation of words as masculine and feminine, neutral linguistics. I know I struggle. I studied Spanish and German and was supposed to be fluent in one of them in terms of scientific literature. And I'm sorry to say I'm not in either, but I can remember enough Spanish to know that we had to keep the A's and the O's for masculine and feminine nouns and adjectives. That transferred into sexual science in the 40s, where there was interest in masculine and femininity in terms of behavior. And now it's become a social construct of male and female differences in terms of gender norms, gender identity, and gender relations. So what do these mean? Gender is not the same as sex. Gender is fluid, and gender norms is how the society has expectations upon an individual based on their sex. Gender identity is how an individual sees themselves or interacts with those expectations put on them by society. And then, of course, we have gender relations of how we interact with one another. So there have been many attempts to measure gender and look at gender in terms of risk assessment for disease. And I'm going to put forward probably a controversial comment and hopefully supported by science, that gender can't be measured because it is fluid. So it changes in these different situations in terms of degree. But what can we do that would give us a handle of how gender influences disease? And that is by looking at the intersection of factors which contribute to gender on the biology. So what does that mean? That means of things that we can physically measure, account for, and actually intervene with if we find that they propose a health risk to the individual. What does that mean? That means lifestyle, your diet, your activity, use of recreational drugs, how you sleep, and your relationships in terms of how much stress your job or so on that you might have. These are things you can intervene with. You can intervene for diet or activity or sleep or recreational drugs. Smoking, for example, is being a risk factor for cardiovascular disease, just for a very common one. And what about the environment? Okay, you all are living in altitude, right? I come up to Denver and I'm like gasping for breath here living in sea level Minnesota, right? Is that going to affect my risk for disease or long-term outcomes? Maybe. Okay, what about people who live in colder climates compared to humid, or the pollutants that you might find in a farm area like I live in, all the factors that go into the soil and weed killers and so on. What about occupational risks? Culture, the attitudes, financial resources that might produce stress or level of education in terms of how you actually absorb material that may say, I need to change my behavior to improve my health. And of course, biases that exist in healthcare in terms of how individuals are treated or their access to care and resources. So I look at this as, yeah, gender does intersect with biology and it's very important. But what's important are which of those components that are associated with gender to put that individual at specific risk and what can be changed in order to reduce that risk or to improve their health. So this is a simple way to look at it. Here are things that we can't control: our genetics, our sex, our age, our hormonal status, and our maybe reproductive status. And part of it we can control, part of it we can't, our history. But then the gender variables, our lifestyle, culture, and so on, are things that we need to account for, which influence health and really can focus on a diverse set of clinical problems which are targeted to the individual. So for terminology in terms of grants and papers and so on, when it's something that's related to biology, like heart failure, I think it's male and female. If it's a risk for gender, men and women is usually the term. A lot of papers mix them, and I know when you're looking at clinical studies, it's really hard to associate that out. But when you're having a particular table for looking at sex differences, if you're looking at sex differences, those tables better be labeled male and female. Or if I get it to review, which I don't get many, I decline papers now, but I still pick out those ones where I know I can make an influence, they better be male and female. So the question that you're all going to say is that, okay, this is all very important and you've gone through a lot of material here. And quite quickly, I was just checking the time here. how can I include sex as a variable in my experimental design? And I think I would take a step back and say, before you ask this question, how to say, how can I include sex in my experimental question or in my research focus? Because if you start with the idea that some of these parameters may not be the same between males and females, then the question then that you're asking should guide your design. So if you're dealing with a sex-specific condition related to reproductive or urological organs, that's kind of, you've got one sex, right? Prostate cancer is not going to occur in females, or ovarian cancer is not going to occur in males, and pregnancy is not going to occur in males. So those are kind of easy, and you can easily justify that. Established cell lines is a little trickier because the longer they're established, they lose their sex chromosomes, and you don't know what you're dealing with, although a lot of information has come out of those. I think in nowadays we need to pay attention of what the sex of those cells might be. And if you have data only on one set of cells, you need to establish cells that are representative of the sex that's missing. So in the context of the research question, when I said you can't say, how can you incorporate it into research design? You got to go back and look at the question. Are there sex-skewed disease prevalence, or is it a sex-specific hypothesis? So what are you studying? And what is the basic unknown within that study, or from the epidemiological data or observational studies that suggest that there might be something different going on between males and females? And I really appreciate that one of the, if you could say, benefits of COVID, it has really raised the awareness that the response in males and females are not the same in terms of susceptibility to disease, hospitalization, and mortality. And hopefully some of that will trickle down into other ways of thinking about disease, especially clinical studies going forward. If you don't think what you're studying is going to show a sex difference, you can pretty well establish that with pilot studies and use that in your grant data or your preliminary data. Randomization is tricky because if you're studying a condition that is sex skewed in the population, you know, the question is, how do you recruit for that study? Do you recruit as it's prevalent in the population? Do you recruit as they come in, collect data from both males and females, and say then that you don't have enough statistical power to look for a sex difference, but you will express the data separately and continue to explore if that's possible going forward. I think you need to report sex and gender components in the measure. And, you know, we take intake forms from clinical studies. Some of this demographic data is already there. I mean, when we look at things that are reported for major clinical trials, they talk about location, they talk about education, income, family status, income, and so on and so on. Some of those things, and diet, some of those things are already there. Always consider your effect size from your preliminary experiments. And you need to look at whether or not there are sex influenced or gender trends, even if you do not have enough statistical power, because this drives hypotheses for the future. So what kind of sex differences you might find, and this is from Peg McCarthy at the University of Maryland. She identifies these as qualitative differences, those which are really distinct between male and female. But then there are also some that distribute different in populations, or that may have characteristics that overlap. But one of the things that I think is most important for us to consider in terms of mechanistic scientists at the levels that you are whether your outcomes are driven by different processes in males and females. Okay, so what's an example of getting to the same outcome but by a different pathway? And one of those is regulation of blood pressure response, cardiac output in response to stress. cardiac output is the result of heart rate and stroke volume. And we have changes in peripheral, changes in pressure and peripheral resistance, which also influence it. In females, this cardiac output is maintained primarily by changes in heart rate, where in males, this is driven primarily by changes in peripheral resistance. So the idea that you have the same outcome, controlled cardiac output, by two different mechanisms associated with autonomic nervous system function, is key to perhaps risk for disease going forward, especially for heart failure with preserved ejection fraction, hypertension, to name just two. So you have these sex biasing factors that are independent, the chromosomes and the hormones, which may actually, oppose each other in order to get reduce variability in the population. You can eliminate one, it may uncover the effect of the others, but keep in mind that males and females may get to the same place, but by different paths. Now, Dr. Regensteiner mentioned when we were talking previously that I was going to talk about the 4-core genotype, which helps to separate these hormones and chromosomes effect. Unfortunately, I didn't put any slides in here related to that, but that might be something that you would want to address in the future. Exactly what is that 4-core genotype, where you can take the sex determining gene off the Y chromosome, put it on an autosome, so you have a female that has testes, and vice versa, that you'll have a male then that has ovaries. So you can separate out the chromosome effect from the hormone effect. I had a question.
Virginia?
Yeah.
I'm sorry to interrupt you. We have a question from Dr. Villa for Dr. Miller. Lane, want to ask your question?
Sure. I guess my question is, when we're thinking about study design, when do you know to go beyond just kind of looking at sex-based differences and categorizing patients based on their age or puberty versus postmenopausal? And when do you know you need to actually look at like hormonal expression levels, especially when you're thinking about these mechanistic type studies?
Mm-hmm.
Or is that?
Yeah, I think, there's a lot of interest now in reporting sex differences, but there's not a lot of interest in reporting why those sex differences are there. And that's where it comes into importance of looking at age, because some of these differences in diseases and so on, the sex differences disappear or reverse actually with age when the estrogen levels start to go down in females. So I think in terms of how those hormones are going to interact with a particular intervention is when you need to start thinking about it.
Okay.
Because there's a lot of do women respond to beta blockers the same as men? If we know that estrogen regulates beta adrenergic receptor expression, and the literature is not necessarily designed to look at that. Mm-hmm. Even if you have the males and the females, because you don't know what the hormonal status of some of those women are. If hypertensive pregnancy disorders put women at risk for premature cardiovascular disease, that's something that's not typically on an intake form for a clinical study. So when you look at individuals that have heart failure with preserved ejection fraction or hypertension, when does that hypertension appear in women relative to their pregnancy that may have had a hypertensive event? Those data aren't available and those studies aren't designed. So you're in a really good position to tease out that next level of when it should be important, because some of these differences need to get into clinical guidelines in terms of preventive measures. For example, monitoring women after they have a hypertensive pregnancy, not when they reach menopause, but perhaps before they get there, right at the time when their blood pressure might go up, not 10 years after it's been hypertensive for so long. Does that answer your question?
Yeah, I think it does. So it's almost more kind of like another layer, more depth. Once we start to know that there are sex-based differences, the next step is kind of the why.
Yeah, and then it's like, so what? I mean, that's where I'm stumbling right now, is because we have all this clinical literature out here and where there are these sex differences. but we haven't seen it transform the clinical practice.
Mm-mm.
We haven't seen it be used to designate thresholds for treatment to be different in males and females. You know what I mean? So like for example, I think I'll just use this, and I don't know if this is true. B-type natriuretic peptide is used as sort of an indicator of risk for recovery from heart failure. But those parameters have been designed for what the level is if you're a male or a female, depending on your age. Because if you set it as a certain parameter and women never reach that parameter, they're never going to be treated.
Yeah.
Where men get it ahead of time.
Yeah.
Does that make sense?
Yes, that makes a lot of sense. Thanks.
Okay, so here's some questions that you can use to guide your approach, because you all want to write grants, right? You want to solve some burning, important question to improve humanity. Not improve your career, improve humanity. If you start with the end of what you want to do, the career sort of follows by. Is there evidence of a sex difference in the disease in humans? And if so, what is it? So I think that this goes back to the question that was asked before. If you know that there's a difference, what is it? And what are you going to do about it? Or what is the question that drives it? Why is it there? How does it influence the disease? How does it influence risk or treatment or mortality? Okay. But, you know, keep in mind, it might not, the data may not be there. And if the data aren't there, you don't know. And so you really have to decide whether or not there's a sex difference or not. And it's okay if there isn't. There may be some things that it doesn't really matter, you know, whether, you know, so I mentioned the coronavirus, but, you know, we get polio shots and for whooping cough and measles and shingles and whatever, all those things, you know, is there a sex difference in those kinds of things that we're getting? But, you know, there might be a sex difference in terms of response to treatment for some drugs associated with depression, renal function for sure. Then you have to consider, is there an experimental model of the condition or the disease? And does that model represent the sex differences observed in humans? And this is where there's been a lot of problems with some of the original studies of genetically modified mice, that many of the genetic modifications were only present in the male mice and not the females. And so much of what is known from some of these experiments, you don't really know what the consequence would be in the other, in the females or the males. And some of it was vice versa too, you know, so have to take some of these experiments with a grain of salt. And when the literature is reviewed, when you review the literature for this, and you have to write a literature review, you know, you just don't say patients, I hate these sentences, it has been found that or whatever, in patients, whatever. What kind of patients? What kind of animals? Why is it there? Why is there differences in studied one outcome compared to the other? Did they have a difference in the sex composition? Was it only one sex or the other? Sometimes it's not mentioned. You have to have it reported. And if it's not, you call that out in your review, as that is a shortcoming of where our knowledge base is at this time. And of course, is there experimental data lacking in one compared to the other? So a lot of scientists initially were balking at the idea of having to include females because they only did males. Okay, now I have to double my sample size, blah, 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 blah. Well, maybe you don't, because if you have a history of studying only one set of animals, if you study some to control for time and conditions of your experiment, you should always include both males and female animals, but you don't have to include maybe not this full complement if those ones that you do now are similar to the results that you derived last year. And it's important to do that because sometimes breeding conditions change, environmental conditions, especially for mice, which will affect their response, especially their immune response. So here's some examples, and I don't know how we're going to do this. We have a few minutes left. Maybe just take a look at these. Excuse me, I was kind of losing my voice. So here's an example of how to account for what to think about. You have somebody studying DNA activation of the immune system. They're studying monocytes, and they want to see, they're treating the monocytes with DNA overnight, want to see what might be released. So how could the investigator improve their use of sex as a biological variable in their study design? Anybody?
Come on, some.
This is not a hard question. This is the easiest one. Remember, every cell has a sex. So you have to account what, identify the sex of your monocytes. You're going to do males and both males and females are going to be mixed. Where's the source of the DNA? How is that isolated? Does it take account the sex chromosomes? These are the things that you need to kind of ask. In an animal study, you have somebody looking at a signaling pathway of in a cancer. They're going to use humanized mice and put the tumors in these mice. It sounds reasonable, but do you see anything here about sex? What's the sex of the humanized mice? What's the sex of the tumor? Does it matter whether they're the same or different or not? These are questions that I would ask as a reviewer, and you should be thinking about as you look at this example. I'm going to skip this one and go here to the human study, just because we're running out of time here. Here's a study that's aimed at looking at a repurposed drug to treat hypertension in humans. They have a recruitment plan for both men and women, and they divided them into younger and older age groups. How can you improve this in the study design? If you were reviewing this as a reviewer, what would you ask? What do you need to know?
In Virginia, we have till 10 o'clock for your session, so.
Oh, so we still have some time?
Yeah.
Yeah, okay. Sorry.
Somebody, you should shout it out. Sorry. Go
I
ahead.
mean, I think for this, again, you need to know, are they going to analyze by sex, men and women? Are they going to collect data on their actual biological sex, or do we know if this is just the gender reported in the medical chart?
Yep.
And then also, are they menopausal, perimenopausal, premenopausal kind of age groups, that kind of thinking?
Yeah. And if they're premenopausal, maybe you want to know if they're taking oral contraceptives, because that could influence the result too. Pregnancy history could be found. And then how about actually recruiting for this study? How do you recruit? Does it matter?
Absolutely.
Yeah, one size does not fit all. I mean, if you wanted to recruit women, sometimes they're reluctant to come into trials because of transportation or care of younger children or older family members, or their risk assessment. Where do you put the brochures? Do you recruit from the medical record and just send it to their house, which is typical Mayo Clinic style? Some other places use more creative ways of putting the brochures in barbershops or hair salons, or gyms and so on. So all those can kind of, you can have a recruitment strategy, but it may not be the same for each of these, the sex or the age of the individuals.
We recruited at bingo parlors for a while to get a demographic.
How did that work? Was that good?
Years ago, it was good. people who went in didn't like the smoke that they also had to breathe in. It did work a while. We mostly use carefully written, we mostly go to My Health Connection. Our health system lets us recruit with permission of the patient's doctors. And we do some newspaper recruiting, but we're very careful because to get who we need.
Yeah, and for studies that are more complicated, you don't want the investigator recruiting individuals. And does it matter if it's a woman woman study coordinator trying to recruit a man or vice versa? Or does it matter? I mean, I think all those things can kind of come into it when you're looking at study design. So I think what we need to remember is that considering sex as a biological variable and identifying a sex difference, is just as one of the questions came up, is not the same as investigating why there was a sex difference and what we're then going to do about it. So if you can kind of keep those two things separate in your mind and say, well, we know that there's a sex difference here, but we don't know why. And so what? If it's there, what are we going to do about it? Or does it influence how we actually behave and treat one another? So I think some critical information that needs to be collected for animal and basic science studies, and all this kind of stuff used to be in the methods, right? When I started out science 50 years ago, we put all this in the methods. Now with methods going to the supplemental material, nobody pays attention to it. But that's one of the reasons why you can't reproduce some people's experiments, because they don't read that stuff in the supplemental material. And some of these things really matter. Culture media for cells, if you have some phenol red in there or other hormones, fetal calf serum, that's those are hormones are gonna be influencing the physiology of the cell. Food types, and these came out of your group there, whether they had a plant-based or a meat-based diet for the rats made a difference in terms of their cardiovascular parameters and metabolism parameters. Life cycle too, we're studying nocturnal animals during the day. Does that really make sense if we're doing rats or mice? And then clinical demographics, which kind of balance some of these things, or instead of a strain, we have race, ethnicity, reproductive status, geographic location, and marital habitation, and so on. These things may be influencing. And, when you look at some of these studies from NIH that go across the nation, they try to pick different locations to say about what happens across younger people or whatever. Most of them are metropolitan areas rather than rural areas. And you wonder whether or not some of these risk factors and things that they're identifying for the metropolitan areas are similar for those in the rural. And the same with, what's going on in Alaska compared to what's going on in Florida in terms of temperature and humidity and so on. Those kinds of things aren't really investigated or considered. Now, we've talked about design. We've talked about what you should consider in your sort of parameters that you measure. Once you have your experiments done, I think we should say something about reporting. So here's typically how many studies have been reported. You have, so your control, your treatment group, and then your outcome. But is this really representative of what might be going on? And if we look at these as what might happen, when you look at your data by sex, you see that these means here are really going to result from these two extremes, which are quite opposite in males and females. So by reporting the data disaggregated by sex, you really gain insight into what's going on with your group, because this difference may not be significant statistically, right? If you just ran the means of these combined groups, it may not be statistically significant. And you just, go on to the next thing. But when you start looking at the difference here, it becomes quite interesting. And you maybe not want to throw the treatment out because, it only works in half of the population. Well, isn't that half of the population important? And doesn't that tell you that we need to take a better look at what's going on in the other half? So I would encourage everybody to report disaggregated data by sex. And, one of our birth scholars, Tracy Weisgerber, called this the never, never zone here, because we don't know what's going on there. Because it's, again, they have the pooled males and females, but they looked at wild type and knockout mice or genetically modified mice, but they actually showed the individual data points. And in here, instead of disaggregating these data by sex, within the genetically modified animals, they identified the males and females by a different symbol. And you can see this also is very interesting in terms of what it might inform your next hypothesis or your interpretation by the results. So the difference in response here may not be because of the treatment effect for the genetic manipulation, but may be the fact that you have a sex difference to begin with. And these are very informative that there's no sex difference. And then you can go on with your experiment and look at the data in a very interesting way. So in statistical analysis then, it's typical to adjust the control for sex. And we will put forward that adjusting for sex is not equivalent to assessing sex as a confounder, because there's no interest in how that sex affects the outcome. So if you're using it to adjust, that means that it affects your outcome, but it doesn't tell you how or why. And isn't that what we really need as scientists at the next level, the how and the why? So if you stratify your data by sex, and this is what I encouraged you to do, this may not be a statistically significant difference, because you may not have enough power to show it, even though you think that there might be something there. The small sample size is going to give you a negative result. But if you look for an interaction term, where you look at the males and females separately relative to the outcome and the interaction of the sex on that outcome, you can see these differences with even a smaller sample size. And it helps then to design your next experiment in terms of numbers, also to interpret the results as the effect of sex of what this outcome might be. I'm going to show you an example here from our own data from our one study we did on hypertensive pregnancy, where we looked at coronary calcification. So this is a CT scan in the heart. The legs are coming out. So this is the left coronary artery, and there's a big hunk of calcium in there. And we looked at women that had a normotensive pregnancy compared to had a pregnancy with preeclampsia. And we individualized the data points for those that had pre-existing hypertension or existing hypertension or not. And here's a good example. When we looked at the mean of these data, yes, the women who had a preeclamptic pregnancy had an increased risk of coronary calcification. But when we actually look at these numbers, there's individual data points. There's a huge, almost not quite half of the women did not. So when you look at this distribution, if these points hadn't been there, you'd say, well, everybody has a risk for coronary calcification if they have a preeclamptic pregnancy. But the question then comes, well, what makes these women different from these? And it proposes then for you to look a little deeper in terms of what those characteristics might be that really put these women at risk for calcification, but these for not. So, there's a lot of things here, what you should do, and we need some tools. And I think you all know about some of these tools. Well, here's the tools for the how to draw the interactive line graphs. And these are free on the internet. And I think many of you probably are implementing these, because I think some journals now are requiring the individual data points to be shown. In terms of training, the Canadian Institute of Health Research was one of the first ones that had some online training. Again, this is free and really quick, sort of 30-minute modules for that. The NIH and the Office of Research on Women's Health are putting these new ones available. And Judy, you may want to comment on these. I haven't been on that website to look at those different courses. That I was a reviewer for some of the material, but I haven't seen the major one. And this particular review, an endocrine review, is a really excellent statistical analysis. And then I'll put a plug in for this book that we look, Sex-Specific Analysis of Cardiovascular Function, goes through the cardiovascular system in detail in terms of sex differences. Judy, have you looked at this NIH material?
I have. I was a reviewer for content as well. I actually think they're pretty good. I'd be really interested in hearing what you think, Virginia, if you want to take a look at them. They're pretty comprehensive and they make you think.
Sure. How long do they take? Can you like go in depth for multiple levels?
You know, what's depth in a primer, but it's pretty in depth actually. I think it take a couple hours, 3 hours.
Okay.
Has anyone done the primer for NIH? And if not, you're supposed to, so please do. But it's nobody has to out themselves as not having done it, but please look at it and give me feedback on how they seem to you as well.
Yeah, the Canadian one takes about 30 minutes per one. I did do those a long time ago. Okay, so what does NIH expect? And NIH expects that sex as a biological variable will be factored into research design, analysis, reporting. I think research design, I would put it into research questions, and the research questions should drive the design. And although we were initially given on review sections this kind of, I don't know, template, guideline, or whatever you want to call it, decision tree about how to score some of these grants. Initially, people didn't know how to score them. They didn't know how to review them. The research community is becoming a little bit more educated in this. And sometimes they think they are, but they're not. And so they ask questions which are above and beyond what the actual study design is. So I think you need to kind of anticipate what some of these weaknesses or the reviewers' comments. Of course, this one, you'll never do this. There's no mention of sex distribution, the topic under med. After this talk, you are not going to do this, right? So you need to think holistically and mention it. Here's another one I've heard. It's unclear who compromises the patient or animal population under study. We talked about that. You better tell them. Give a good description of age, sex, and hormonal status, and provide the rationale for these decisions of inclusion or exclusion of your experimental groups. There's no mention of how recruitment will be balanced by sex. This is important, especially for conditions that may show, human studies that may show sex difference in presentation or outcomes. You need to tell them what that is and go into detail. A study is underpowered to identify sex differences. You can easily have power analysis, or say it's not designed to study sex differences. If it's not, that you're going to do a secondary analysis to look for that hypothesis in the future. There's no mention of the statistical section of how the data will be analyzed. All this, there's no excuse for this. You have to tell them and say for sure that you're going to look to see if there's a sex difference, it's powered, and if not, we'll do it in the future. Now, I'll give you a good example of that. We had one of our birth scholars was studying spontaneous coronary artery dissection, and she was doing some imaging as people came into the emergency room. Well, of course, it has a big bias for women compared to men. So she was impossible for her to look for sex differences for the parameters that she was, although she knows they're there based on the number of men she was going to be able to recruit. We subsequently worded this such that she takes all comers, she collects the data from the men, knowing she's not going to have enough power to be able to identify a difference, or even enough men at one time to show anything. And but over the course of the grant, she should be able to accumulate something to formulate a hypothesis for the future, and it seemed to work as she got the grant. So let's review. I have a few minutes left, and then we can have some questions. So this is easy. Let's go back and say, you're supposed to be able to say why it's important to consider sex as a biological variable in research design. Anybody want to answer this question? This is a test. You can't feel this in front of Dr. Regensteiner. Okay. So somebody has to answer this.
Please answer it.
Oh, come on. Did they all go away?
No, they're here. Come on, somebody. Why do we care about sex as a biological variable? Natalie, I'll call on people.
Sure. I mean, we want to understand sex effects. We want to understand, you know, differences in outcomes or treatment or, you know, impact by sex among many other reasons.
Yeah.
Thank you.
Because it's the essence of holistic medicine. How's that?
I like that.
Personalized medicine. Why are there sex differences?
Because of this is one of my areas, but because of all the things you mentioned, sex chromosomes, hormonal milieu, gonadal status, puberty. I'm a pediatric endocrinologist.
Right, genes and hormones and the environment. We have one of your scholars is passing here. What is the difference between sex and gender?
Sex is the biological, the XY chromosome, and gender is kind of everything else that incorporates the external influences on it.
Yay, very good. Sex is biology, gender is everything else. What are the key components to consider in developing a research strategy?
Mary Whipple.
I think you mentioned sex as well as age, hormonal status, thinking about the role of these issues in both study design and for analysis, in recruitment efforts as well.
Yeah, we got those. And then the questions, and materials, the results, and the reporting. Very good. Your scholars are on their way.
They are.
And with that, I'll just say thank you very much for allowing me to interact with you all today. It was fun. I hope it was worthwhile, and go forth and get those grants. Champion sex differences. It's now more important than ever.
I have a question for you, Virginia.
Yeah.
Thank you for your wonderful talk. I really learned extensively myself, and I appreciate it. And I want to ask you, are you aware of any guidelines, any sex-specific guidelines that are in practice that are used in clinical care? Because I'm actually writing a paper where that's part of it, and I don't see any.
No, I mean, one of the things that we've seen change in clinical practice, and that's not necessarily a sex-specific thing or a sex difference thing, but we've seen changes in clinical practice relative to women having bilateral oophorectomy for benign conditions before the age of natural menopause. Those kinds of operations have declined, and it's now clinical practice to give them a hormone if it's needed to, and they're not genetically susceptible to other conditions, to give them hormonal replacement at least until they reach the age of menopause. The clinical guidelines for hormone replacement have been all over the place. I worked with a fellow in emergency medicine, and there's now the initiatives to change when women come in for emergency settings in terms of the parameters of which some women with some cardiovascular diseases are referred for imaging and interventions that not before. The American Heart Association, with their Keep with the Guidelines, have changed the parameters to increase the medication right away for women with cardiovascular drug reducing or risk reducing drugs. But that's probably that's all what I know. I mean, how it has affected other treatments in terms of osteoporosis, or statins are still given out like crazy. And I don't know that that's necessarily good for women as for men, because their cardiovascular risks in terms of lipid metabolism is different. So I don't know, Judy, that's a really good question.
Does anyone else know of any sex-specific clinical? We have people here in endocrinology and emergency medicine in various areas. Yeah, I didn't find any. I didn't find an example where somebody go into a clinic and get a different type of treatment based on the fact of their sex.
Yeah. I mean, you know, some clinicians may do that, but it may not be in the guidelines.
Mm-mm.
You know, that's kind of where you run into this sort of art of medicine thing, where some of it is like, I hate to say it, kind of experimental, but if this medication doesn't work, let's change it to this one or whatever. And although some of the drug interactions are available on some of the FDA website things for side effects and so on, it may affect what first line medications might be for some depressive treatments or things like that. But I don't think it's followed through for like insulin resistance, metformin, or, you know, insulin treatments. I don't know in terms of metabolism how that would work.
The only drug for which I'm aware of a sex difference in dose is Ambien at this time.
Yeah. And some people were saying that that's not true, because, you know, it shouldn't be a sex difference because it's related to body mass and fat mass and so on. But I don't think that's the case, because when you look at the metabolism of the drug, the pharmacokinetics are very different between males and females. So,
Is it possible for us to get your slides, Virginia? And if not, that's fine. There's some
yes, it is. The problem is they might be too big for me to send. I may have to put them in a zip file or something, which I'll have to figure out. One of the things about being retired, not only did I lose my administrative assistant and can't keep my own calendar anymore, but I've lost my technological expertise from younger people that are around me all the time. They can do this in their sleep. It'll take me a couple hours to figure it out.
Oh, sorry. It's just you have some
That's okay.
lessons are on there that I think are fundamentals that would be helpful for people.
Yeah, that's fine. Yeah. I'll try to get them to you sometime before Monday. How's that?
Thank you. Very nice.
Yeah.
Any other questions for Dr. Miller? This is just such a rare opportunity. I am reveling in this opportunity, so I just can't thank you enough.
No, it's fun. I'm glad to be able to do it, and I'm glad it was useful to you.
Highly so, and nice comment in the chat.
Yeah, good. All righty.
Okay.
Well,
Thank you.
Thank you.
Thank you.
Okay, you're welcome. Have a good day, everybody.
You
Stay
too,
cool.
Virginia. Thank you.
Okay,
Bye-bye.
bye-bye.
Thank you. Thank you, Dr. Miller.